So this is part 2 of 3. This is going to go over the observations for and conclusions regarding organic evolution. In the classroom, I take a lot more time on this. I tie it into Charles Darwin, and we'll definitely talk about him a little bit, so you can see why he's such an important figure in all this. But these initial observations that we have on this first slide here are things that hopefully you've taken away from the class so far this semester. Sedimentary rocks are deposited in layers with the oldest on the bottom. We can do correlation. We've done that in class. From that, we developed the relative time scale, and we learned that the Earth is very, very old. And this is something that Charles Darwin was kind of on the leading edge of in the early 1800s when he went to college. This is one of the new areas that was being studied in school, and something that he studied in particular. He worked with some of the earliest geologists who were studying sediments and defining these terms in this form of study. So along with that, we have the fossil record embedded in those sedimentary rocks. And when we look at the fossil record, we find that younger rocks have more organisms similar to those living today at all levels, from species up to kingdom. And the fossil record includes appearances and extinctions of many, many species. When we look around the planet, one of the things that we find is that many organisms are similar but unique. And this is, again, where Darwin comes back in. He certainly learned about the fossil record. And when he got out of college in the, he was born in 1909, so it's the 1930s now, or I'm sorry, the 1830s. He was born in 1809. He gets out of college in the 1830s, and he's looking for something to do. And one of the options available to him was to go on this voyage of a ship called the Beagle. He was to be the captain's companion because they were both upper class. And he was to go around the planet on the Beagle and collect samples and send them back to England for observation. And so what he was able to do is he observed this. He was one of the first people to really get the impact of this. No matter where you go, you find organisms that are similar but unique. They're different species, clearly, even though they have some similar characteristics. They're typically confined to specific areas, and when you go to different continents and islands and things, you see different assemblages of animals and plants. And this also includes fossil organisms. So here's the voyage of the Beagle. You'll notice it took 5 years. It started in England. It went down to South America, to the Galapagos Islands, Tahiti, around Australia, through the Indian Ocean, along the coast of Africa, and ultimately back to England. And all along the way, Darwin is collecting not only samples of living organisms, but also fossil organisms, and sending them back to England. So what does he find? Well, an example is from South America. He finds a number of organisms, both living and fossil organisms, that are different from what he was used to in Europe. And notably, when he looked at the fossil organisms, they were very closely related to the living organisms. And so there are 3 pairs of organisms here. The extinct organisms are things that Darwin found in the fossil record, and the living organisms in all 3 cases are the smaller organisms, are very closely related to the extinct organisms. And these are things that you don't find in Europe. These are organisms that aren't there. So what he's seeing is there's a history here. Not only are the organisms different, but they've been different throughout time. He goes to the Galapagos Islands. One of the things that he does there is collect finches. So what he did was he murdered the birds. He actually collects the birds and Um, he stuffs them and sends them back to England. He thinks these are different species. When he gets back to England, he finds out that they are not. The friend of his he sent them back to said, "Hey, where'd you get all the finches? There's a lot of variety here." And that really gets him to thinking about why there are so many different kinds of finches on the Galapagos Islands and there aren't a lot of other types of birds. So after he gets back to England, he studies not only finches, but he starts studying lots of things. Um, he studies embryos. He studies a variety of organisms and looks at their anatomy and physiology and compares them. And since then, we've done even more studies. And what we find is that the cell structure of living organisms is similar, no matter if you're looking at a plant or an animal or a fungus or just a single-celled organism. When you look at embryos of mammals, birds, and reptiles, they're all very similar through their early developmental stages especially. When we look at organisms, we find things that are called homologous and vestigial organs. So your internal structure is very similar to the internal structure of other mammals, as well as other warm-blooded animals. Last week we did some cladistic stuff. Homologous organs definitely play a role when you're trying to figure out the relationships between organisms. It's a similar organ, but it has a different function. And we'll take a look at some examples in a few slides. A vestigial organ is similar to a homologous organ, but a vestigial organ is an organ that we don't see an actual purpose to. So for example, you have a few bones fused together that we call a tailbone, doesn't seem to serve a significant function in humans. So what does this look like? Well, these are homologous organs in fossil and living organisms. So it shows the forearm of a person and a dog. And it also shows the same forearm in several different types of fossil organisms and how those bones look. And if we want to look at them more closely at your forearm, there's a human arm on top, and we've got dinosaurs, birds, pterosaurs, bats, penguins, lizards, all different kinds of what we call quadrupeds, which are animals with 4 limbs, and they have the same general structure in their limbs. They have one big bone, and then in front of that they have 2 thinner bones, and then they have a lot of little bones that make up their hand or their fin. So these are what we refer to as homologous organs. This is an example. This is a picture I took of a fossil of a modern whale. And one of the things that you'll notice on here is you notice its forelimb. There's the big bone, and then the 2 smaller bones, and all the finger bones. And also, there it is up close. So you can see that forelimb looks very much like a forearm and hand. It's just the lengths of the bones are different. And at the tail of a whale, what you find are 2 tiny little hip bones, and these are what we would call vestigial organs. So whales do not have back legs, they don't have hips, but they still have small hip bones. And there's another picture of those hip bones. This is from the book Your Inner Fish, and this shows you the skeleton of Tiktaalik. It shows you basically some of the structures that we find in a modern tetrapod, which is at the bottom, a fish, which is at the top, and Tiktaalik occupies a space anatomically that is somewhere in between. These are some fins, again, more homologous organs. They kind of show you how we go from the fin of a zebrafish, which doesn't look much at all like your forelimb, but through a few stages, you get to this bottom fin, acanthostega, which looks kind of like a forearm and hand. There's our big bone, our two little bones, and then our lots and lots of finger bones. And there's the bones from the front fin of Tiktaalik, which is our transitional organism. Now, Darwin didn't know anything about Tiktaalik, but he speculated that such an organism might be found in the fossil record, and indeed it was. The other thing Darwin didn't really know about was genetics, and one of the big tests for evolution, the idea that all life is related, was when we discovered DNA and RNA and found out how it worked. And it turns out that the DNA and RNA in your cells is very similar to the DNA and RNA in all living organisms on the planet. And it even controls similar areas of body development in everything from a human to a fly. We then bring in cladistic analysis to get a better understanding of how these relationships work from a statistical standpoint. And we come to the conclusion that the characteristics of populations of living organisms have changed through time. And this really is the core definition of organic evolution. Life has become more complicated, it's become more diverse, and all life is related. And at this point in the sciences, organic evolution is not even really a theory, it's accepted as a factual observation. We look at the fossil record, we look at living organisms, and it's really difficult to go forward with the study of biology, medicine, or fossils without simply accepting the fact that organic evolution has occurred and continues to occur. Now, the theoretical part of organic evolution is looking at the patterns. We look at how one species can form from other species, how they become different. That's what we refer to as divergence. We look at how things become extinct and other organisms come in and take their place. The other kind of interesting trend that we see is what we call convergent evolution, where organisms living in a similar environment over time very often develop similar strategies. So the fin on a fish and the fin on a whale are actually, anatomically speaking, quite different. But the general form of the fin and really the organism itself looks very similar. So that's what we would refer to as convergent evolution. And we'll talk more about phylogeny a little bit later, but well, actually, you talked about it last week. It's the conclusions that we draw from cladistic analysis. What's related to what? So those biological family trees that we develop are phylogenies. And this last item on here, the gradualism and the punctuated equilibrium, is another theoretical argument that we're having within the community that studies evolution about exactly how does evolution progress, gradually or in little fits and starts. So the other concern that a lot of people have is what about missing links? And I think that taking a look at those slides on Tiktaalik show you exactly how that works. We have found many, many missing links. We find many fossil species that help us make the connection between two different groups of organisms. However, the fossil record is spotty. So whenever you find a new fossil that fills in a gap, then there's a new gap before and after that fossil. So missing links is useful, and we do look for those linking organisms, but it's also kind of a strange concept because it's a problem that you can never completely solve.